Good morning to everyone and welcome. So I'm going to explain the research that we do here at the Josep Carreras Leukemia Research Institute in Barcelona in Catalonia. So I will start with this slide that describes a little bit about identity and what's epigenetics. As we mentioned before, some interesting points about the genetics, and it's relevant to remember that all our cells, they have the same DNA sequence, but they do different things. The eye, the gut, the brain, and this is not in the DNA sequence by itself, but in the regulation of DNA. And this is what we call epigenetics, whatever is over taking care of genetics. And in this regard, there are several marks that we call epigenetics, but probably they're summarized in a couple: histone modifications and DNA methylation. DNA methylation is very easy. It means that the C, instead of a C, can be a methylated C. And this is like a switch off of a gene. It's like a brake in a car. It stops or silences gene expression. This is relevant because, for example, when we look at differentiation, we have a stem cell and we generate any tissue. We're changing the expression of thousands of genes. And this is associated with the change in epigenetics of many candidate targets. And in hematopoiesis, when you move from a hematopoietic stem cell to a fully differentiated karyocyte, neutrophil, T cell, you change the epigenetic patterns of the cells. And in fact, mutations in the epigenetic enzymes, like DNA methyltransferases, are able to block the differentiation status. And they are associated with leukemia, lymphoma, and many other tumor types. There is another process in biology, apart from differentiation, that is called transdifferentiation. It means you have a cell status and you move to another cell type without going to this intermediate stage of a stem cell. This is a way that cancer uses to escape from therapies. For example, a B acute lymphoblastic leukemia can become an acute myelogenous leukemia under the pressure of CD19 inhibitors. It's a way to escape. They changed the clots and they became a different cell. A prostate carcinoma responds to hormonal therapy, becomes now a neuroendocrine carcinoma that does not respond to therapies with androgenic therapy in this case. And we have shown that this process is related to changes in DNA methylation. Just to summarize here, red, it means methylated, green and methylated, and you see that arrow that it's pointing at a particular methylation site that change upon transdifferentiation. Maybe the most famous thing about epigenetics in cancer is that there are many tumor suppressor genes that they are not mutated in a somatic manner, but what happened here is that they are hypermethylated. And this DNA methylation silences these good genes, these anti-oncogenes, these tumor suppressors. And this happened in all these pathways: cell cycle, DNA repair, angiogenesis, apoptosis, cell adherence, etc. And this it's in part what makes these hallmarks of cancer. The hallmarks of cancer by Dr. Hanahan showed different features that characterize a tumor cell. And in the last classification, after many years, outside Scott, has been included epigenetic reprogramming. Now it's part of the hallmarks of cancer. But in fact, Doc did this wrong because epigenetics is everywhere. It's the same that genetics is not one part of the puzzle, it's everywhere. It's a mechanism of controlling gene activity. So epigenetic defects affects all pathways. And to give you an example of a pathway that is targeted by epigenetics, this is one type of apoptosis that has been mentioned now in the previous talk, that is called ferroptosis. It's a way of dying of the cells, physiological, associated with iron. And what happens is that cancers, to protect against ferroptosis, we have one pathway, glutathione, and we have one enzyme, the ferroptosis suppressor 1, that protects against ferroptosis. What happens in leukemia is that this gene is silenced by DNA methylation, and then all this avoiding ferroptosis depends on this pathway. And now we can target this pathway with candidate drugs for ferroptosis like this, and these cells, leukemia cells, die faster. This is an epigenetic vulnerability of tumor cells. You can think that this is complicated, but this can be more complicated than this slide. For example, it's not only changes in the DNA sequence, not only changes in the DNA modification, like DNA methylation, but there's another molecule, RNA, that can be highly modified. Many chemical modifications can occur in RNA, not only in DNA. This is much richer than DNA. For example, you can have methylation of RNA at the cytosines or at the ACE. You can have a pseudouridine instead of uridine in the RNA. And you have a lot of enzymes regulating this activity. Just to mention that in transdifferentiation and differentiation, there are changes. You're going to see it in M6A RNA. This is a mark that regulates activity of RNA. Okay, so in the last years, my lab has identified many defects in the enzymes that regulate RNA modifications in cancer and depends on tumor types. For example, enzyme 5, it's an RNA cytosine methyltransferase that is lost in brain tumors, in gliomas. We have TRID1, that is an RNA modifier of transfer RNA, of tRNA, and this is gained too much activity in a small cell lung cancer. And you have all these types of changes. But talking about, you want to focus, for example, on leukemias or lymphomas. This is one example of pathways. If you remember from the School of Medicine or Biology, RNA, to be stable, you need to add a cap at the beginning of the RNA. And this is highly regulated. What we identify is that some leukemias, they have lost an enzyme that regulates the cap of RNA. And this changes the stability of messenger RNAs because it's methylated enzyme that regulates that cap. And it behaves like a candidate tumor suppressor. This is when it's lost, enhances tumor growth. When you restore activity of these enzymes, it inhibits the growth of the leukemia. And this enzyme is targeting, for example, messenger RNAs that regulate MYC. And MYC is one of the most powerful oncogenes that we have there. Okay, it's in many pathways. It's pleiotropic, it's multifactorial, it's everywhere. Another example I want to mention is that very recently we identified that another enzyme is called ALK-BH3. This is regulates methylated 1A in RNA. This one enzyme is lost in lymphoma. And it's happened in many types of lymphoma, with a particular type in Hodgkin lymphoma. What are the targets of this enzyme? We have done what is called an RNA immunoprecipitation with an antibody against this mark and find targets regulated. And we have seen that this enzyme regulates collagens. And it's very interesting because you don't expect a lymphoma to express collagens, to have collagens around. It's other tissues, not a lymphoma. But there are around 10–20% of lymphomas that they have this extracellular matrix, very rich and with an excess of collagens. And these are the same lymphomas that they have methylation of this enzyme. What's interesting is that these lymphomas are really the lymphomas that do poorly. The good news in Hodgkin lymphoma is we're able to cure 80–85% of these lymphomas. But there are 15–20% that are resistant to therapy. And these 15–20%, almost all of them, they have methylation of this enzyme. Because this creates an extracellular matrix that takes resistance to many drugs. So this is a summary of these results here in this slide. You can read some about this story about this field can be called by analogy epitranscriptomics, epigenetics in DNA in RNA epitranscriptomics. And you can read this, for example, in this review that we did for cancer discovery. I want to now move to the contribution of epigenetics to immunotherapy. It has been mentioned also before. This is an emerging field, how we can treat cancer, just enhancing the immune system. And this is important because we know that not only cancer cells, but also the immune cells around, the T cells, the B cells, macrophages, they have important epigenetic changes in the disease. And this contributes to the immunology in the niche, in the tumor. So one question that we had at the beginning was the following. As you know, immunotherapy, like immune checkpoint inhibitors, has they represent a great advance in the treatment, for example, of non-small cell lung cancer and melanoma. But only a few patients respond to therapy, and this is still expensive drugs. So can we select which are the responders here? And here what we did is we took a picture of the DNA methylome of non-small cell lung cancer. We are experts in using DNA methylation microarrays in humans and in mouse and other species, and looking at responders and non-responders to anti-PD-1, we identified that the signature that we call epi-immune is a good predictor of response to immune checkpoint inhibitors. If you are epi-immune positive, and at the same time you are PD-L1 positive, you have an infiltration of CD8 lymphocytes and a high mutational load, this is going to be a great responder. This is the best responder you can have. Everything is negative, there's no need to give the drug. There will not do response. It's a waste of money and time. So this is an example of these biomarkers, in this case, epigenetic biomarker. We have translated this type of knowledge to a new therapies. New therapies like CAR T-cell therapies. This is probably the newest kit on the world. This is using cells, cancer cells, using our cells to defend against the disease, the cancer. This has been exploited mostly against treatment in B-cell leukemia and B-cell lymphoma. So we have T cells from the patient, we make these cells really, really mad, and they go against an antigen, in this case, CD19. And this was the first patient, Emily Whitehead, and now it's 2020, 22, she's fine after all this time, so this is great news. A patient that was at the verge of death here at that time in 2012. So this is great, but there are questions. Question is very expensive, extremely expensive treatment, CAR-Ts. Second question can be adverse effects, neurotoxicity, cytokine release syndrome, for example. And the third question is that there are primary resistance and secondary resistance to the treatment. Can we have ways to predict the behavior of CAR T cells? So what we did is a collaborative study with different hospitals in the world and analyzing the CAR T cells, taking a look at the epigenome of CAR T cells to see if we can predict, if that epigenome can predict the behavior of those T cells when they are transfunded, when they are given to the patient. Using this approach, we identify a signature that we call EpiCAR, that if these cells are EpiCAR positive, it means they are going to have a good clinical response when infused to the patients. These cells, they have a high level of fitness. These are very good cells to be put back in the patient. What are these cells? The best cells, CAR T cells according to epigenetics. The EpiCAR cells, this has been validated by flow cytometry, for example. What they have is they have a high population of naive T cells and central memory T cells. They are more immature cells. For instance, that's the reason they last long in the patient and they have longer effects in this patient. If the CAR cells are enriched in effector memory T cells or effector T cells, they have really a lower response. Probably these cells are too much committed and they cannot stay longer in the body of the patient. So epigenetics is providing a readout, in fact, of differentiation of those cells. Apart from this, it's relevant to mention that when you have a mutation, more or less you are done. You can try to block the mutation, but epigenetics is more dynamic. came in and came out. It's very dynamic, Mark. So there are all these drugs that we call epigenetic drugs that blocking these enzymes can revert the epigenetic marks. And some of these drugs, 9 of them, have been already approved for the use in patients in oncology. They are approved for the use in leukemias, in lymphomas, and sarcomas so far. Here we have one of the first patients that was treated with an epigenetic drug, an inhibitor of DNA methylation, and he had an MDS myelodysplastic syndrome. It's a form of pre-leukemia. And these drugs, what at the end are doing is inducing a lot of differentiation of the cells. I would say that they're more cytostatic than cytokillers. These are cells that block the cells that grow, but they're not killers. So they're blocking there. They're staying there. They're good. They're frozen in time. You're frozen in time 20 years, we're fine. But they're frozen there. And the second example is drugs in cutaneous lymphoma. You can see that the patient there, that is not psoriasis. That is the lymphoma that's happening there. And you see that the good effect that they have. It's not only time of life, but quality of life in these patients, because these drugs are very well tolerated, even at very low dose, maintaining time. This is not like a shock of the chemotherapy. It's very low dose, maintaining time. And these are the different drugs, and they are in development. As I said, none of them are approved. And importantly, now there are clinical trials combining epigenetic drugs with immunotherapy. The idea here is to awake different antigens, and our immune system can recognize more targets. Okay, and these are there are different clinical trials now. Talking about new drugs, this is a drug that we developed in the lab. It's an inhibitor of histone deacetylase number 6. And this drug is very specific of that protein, and it's having a lot of effects in lymphoma. Is our main target. And in lymphoma, particularly in what's called mantle cell lymphoma, that is a very bad lymphoma. Lymphomas that some are very good and some are pretty bad. This is a mantle cell lymphoma, very few options of treatment of this tumor type. You can see here in the next slide that these drugs, what they do is, in this case, they induce acetylation of target proteins. And you see below, this is the nice experiment there. So you see in red, green, the cyclophosphamide, that is the drug from the old times still given to the patients today. Is that the standard treatment of these patients with this drug, with this tumor. And our drug has the same effect that the classical drug, and it's pretty well tolerated. And now has started a clinical trial using this H-Stack6 inhibitor in lymphomas. And hopefully we'll see the results in the coming year. I would like to mention that this wall of epitranscriptomics will produce drugs also. The same that epigenetics has given now 9 drugs approved. There will be drugs approved from epitranscriptomics. And the first drug that maybe will get approval will be a drug that it's an inhibitor of M6A RNA methyltransferase. Okay, there's different companies working on that, and we have shown that this drug is powerful in the impairing transdifferentiation. So maybe it's involved in inducing differentiation of some cells. Okay, and it will be work in a very similar manner than epigenetic drugs in our hands. I would like to finish with just a few concepts now about single cell, because this is a new technology that is going to change the game a little bit. It's like when DNA sequencing started, or deep sequencing, or even PCR started. So now we can look at detail at one cell among millions of cells. And this is something we're doing in our lab, in our institute, and if you want to collaborate with us, I'm more than happy to provide you with this technology at the Josep Carreras Institute. And these are Marta and Nacho doing this work. So this is a technology that we have. So we can measure RNA expression of one cell among many others using 10x. We can measure one cell in a paraffin section among many others. So you can see the microenvironment, what's happening there. You can imagine like this, like a multiplex immunohistochemistry, but 1,000 transcripts. And it's very nice pictures there. And we can do also single cell DNA sequencing and single cell protein. And this also are very relevant information. for example, to study relapse and where is the resistant cell at the beginning. And I want this, of course, in hematology and oncology has many applications. I want to give you one example of a study that we're doing here using single cell. As I mentioned, in MDS, myelodysplastic syndrome, you give an inhibitor of DNA methylation, and this works many times, but sometimes does not work at all, or just work partially in these patients. And there is no way to predict why this drug in some patients works and in others don't. So what we did is we did a single cell analysis looking for single cell mutations and single cell protein expression in these patients, looking at the moment of diagnosis of the disease and when they finish the treatment with a hypomethylating agent, comparing both time points. And this is the type of results we're obtaining. This is what's called fish plots. The fish plot is a way to analyze mutant clones. And on top, you have a patient that underwent complete remission. You see that at the beginning, this patient has 3 mutant clones with different mutations. When it finishes the treatment, at the end, there's nothing there. It's not true. There is one cell. We saw one cell. It's there, but almost nothing there. So this is a patient that has complete remission. And below, you have a patient that at the moment of diagnosis, it has 2 mutant clones. After finishing the treatment, you see that there is a reduction of the clones, but the clones are there. They are reducing the size of the clones. So this leads to partial remission. And we have patients that have no response. No response, the clones, mutant clones, did not care about the drug at all. There's no effect here. And there is even patients, no responder, that are worse than this. Are there patients that take advantage of the drug? The drug is selecting mutant clones. I think this patient at the beginning has these 3 mutant clones, and after giving the drug, you see the emergence of new mutant clones with new mutations. It's a Darwinian selection that we don't know why, but it's happening there in this case, that do not respond to a therapy. As I said, we can combine this with proteins, and we can analyze, this is like, it's called a human representation. You can combine, in this case, mutation and protein at the single cell level. And this is a patient that responds to treatment. And you can see, for example, in orange, you see these cells. These cells, they are hematopoietic stem cell progenitors. These are the stem cells that carry the original mutation in MDS. You see that after finishing the treatment, these cells are gone. You don't see the orange below, but you don't see also the red, because the red are the mutations. What are the mutations? So everything became blue. So you have less hematopoietic stem cells, and you have less mutations, or no mutations at all. In this case, this is a complete responder to the treatment. But you can have the other scenario, the bad scenario. This guy, for example, here this patient is a non-responder. You see the orange cells there, hematopoietic stem cell progenitor. After treatment, they are there, do not care about the drug, and the mutations in this case has reappear and emerge new mutant clones. This is that patient I told you before. It's a patient that has new mutations happening in these different clones, among them in hematopoietic stem cell progenitors. So I think this can be a good way to measure what's going to happen to these patients. If you have a picture at the end of the treatment, we'll have some ideas about what's going on, what's expected to happen in this patient. And probably new targets, because some of these new mutations can be targeted now by other drugs. So I would like to finish here, really. I would like to mention that this work that we are doing in our society, in our neighborhood, in Barcelona and around, can be translated to anywhere in the world. And because many of the diseases that we're seeing are seen also in this part of the world. There are particularities, many of them associated with the environment and with virus that you have around here that we'll have others there. But I think that together we can learn a lot, and we're happy to help in this regard. I would like to finish here and thank all the members of my lab and all our supporters. Thank you very much. And if there are questions, more than happy to answer.
I'm very impressed. Yes, I probably say obvious thing, but it is beautiful work. It is the first part is beautiful, and of course the single cell sequencing protein and genomics you are using this stuff even now for
Yeah.
but the first part was beautiful with it. Really, this is new area, and you are really advanced. I'm just making comments because I'm not in this area, but definitely is huge potential for any kind of
Absolutely.
stem cells or whatever this is, right? Thank you.
Thank you very much.